turquoise, magenta, orange, violet. The names we give to colors are as varied and rich as the spectrum of color itself. They are a description of the interaction between objects and light and the human observer. In the realm of color, the language we use to describe colors and the differences between them has become increasingly important. Yet language is often inadequate when we attempt to describe the subtlety and sensation of color. This object is blue, so is this one, and yet they are not the same blue. What terms can we use to describe the differences between these two similar but different colors? How can we measure color difference? And why is it important? The quality control of color is an essential step in the manufacturing of a wide range of products. Matching the color of a new batch to an approved standard requires highly skilled visual assessment. But visual assessment alone is not enough. Different observers do not always agree on a color match. Even a single observer may see the same samples differently at different times. For while the human eye and brain are quite sensitive in the perception of color, they are not consistent. To accurately match color and minimize the possibility of customer rejection, we need a way to objectively measure color and color difference. The concept of color measurement begins with the way we define the sensation of color. Color sensation can be described in terms of three different aspects: hue, chroma, and lightness. Hue relates to whether a color is red, yellow, or blue. Chroma relates to the intensity of the color, or how much of the color we see is gray and how much is the saturated color. Lightness refers to the lightness or darkness of the color on a scale ranging from black to white. These three properties represent the three dimensions of color. These three dimensions can be described by a color's tri-stimulus values, X, Y, and Z. The tri-stimulus values are a product of the way a sample interacts with light, the light source under which the sample is viewed, and the way our eyes react to light. By describing each of these elements numerically, the spectral curve of the sample, the spectral power distribution of the light source, and the standard observer data which describes the eye's response to light, we can combine them and determine the total amounts of the standard primaries needed to objectively describe a color. The amounts of the red, green, and blue standard primaries that make up a color are represented by the tri-stimulus values X, Y, and Z. These values are difficult to use in a practical way, however, unless we have a map of color. One type of map is the chromaticity diagram. It provides a graphic description of color space. With this map, we can plot any color by means of its chromaticity coordinates. The chromaticity coordinates are referred to as small x, y, and z. They are determined by dividing a color's tri-stimulus values, capital X, Y, and Z, by the sum of all three. These tri-stimulus values are calculated for illuminant D65, 2-degree observer. Only two of these coordinates are needed to specify the hue and chroma aspects of a color, so the CIE, the International Commission on Color, recommended the use of small x and y. To use the x and y coordinates, we need to add x and y axes to the chromaticity diagram. The x coordinate for our sample is 0.2209, the y coordinate is 0.2409. The outer edge of this color space is determined by the small x and small y coordinates of each wavelength of the visible spectrum. The violet end of the spectrum at around 400 nanometers and the red end at around 700 nanometers are connected by the purple line. The purple line represents the non-spectrum colors. All real colors plot inside the chromaticity diagram. The hues of the spectrum can be seen along the curved perimeter. Chroma increases outward from the neutral point near the center. As we have seen, we can plot the hue and chroma aspects of a colored sample in terms of its chromaticity coordinates. The likeness of the color, its third dimension, can be shown through the use of a third axis. This axis is perpendicular to the x and y axes. A third coordinate determines the color's position on the axis and represents the likeness of the color. This coordinate is actually the color's y tristimulus value and is referred to as cap y. A cap y value of 100 is a perfect white, while a value of 0 is a perfect black. Our sample has a cap y value of 33.36. A color can now be defined by its small x, small y, and cap y values. If we use these values to plot the location of a second colored sample, the direction and distance between the two samples will represent the difference between the colors. The position of the two samples also tells us something about their visual relationship. Samples which plot close to one another will be a better visual match than samples separated by a great distance. The chromaticity diagram offers other practical uses as well. We can describe a variety of light sources and plot their locations on the diagram. To do this, we need to first understand the concept of color temperature. Color temperature is determined through the use of a light source known as a black body. The composition of a black body may vary. For the purpose of illustration, a graphite block with a deep hole or cavity can be used to demonstrate the concept. Light entering the hole is absorbed by the walls of the cavity so that the hole appears perfectly black. As the block is heated, the walls of the cavity release radiant energy, which we can see escaping through the hole as the temperature of the black body increases. With increasing temperature, the color of the escaping light changes. Because this color is dependent on temperature, we use the term color temperature to describe the color of the light emitted by the black body. We can also use this term to describe the color or chromaticity of a light source. Color temperature is expressed by the term Kelvin. A tungsten lamp with a color temperature of 2854 Kelvin emits light with the same color appearance or chromaticity of a black body heated to 2854 Kelvin. D65, an illuminant representing daylight, has a color temperature of 6500 Kelvin. Its chromaticity is equivalent to that of a black body heated to 6500 Kelvin. Just as we can measure the color of a sample, we can measure the color of a black body at various color temperatures. We can plot these colors as a string of points called the black body locus.
We can see that the chromaticity diagram is a useful way to describe the difference between common lamps. The diagram has many other useful applications. Right now, however, we're going to talk about one of the deficiencies or apparent deficiencies of the chromaticity diagram. If we were looking at two red samples, these two samples might be very close, a fairly close match, and we see a small difference. Our brain could assign a difference of 1 unit to these two samples. Now, if we measure the two samples and plot them in a chromaticity diagram of some scale, we'd see that this arbitrary 1 unit is represents about 1 inch difference in the chromaticity diagram. We did the same thing with two blue samples, and when we looked at these two blue samples, we see that they also are not an exact match, but they're different from each other, about the same as the two red samples. If we measured these blue samples and plotted them in a chromaticity diagram, we may find that they're only 1/4 of an inch apart. And if we did the same thing with two green samples, they might be 2 inches apart. Then we'd realize that from the standpoint of perception, that this diagram is not uniform. The same distance between two samples in one part of the diagram does not represent the same visual difference in another part of the diagram.
In 1942, David McAdam documented the responses of the human observer to small color differences using the chromaticity diagram. McAdam began with a point which represented the color of a standard. Based on visual assessment, he then determined a series of points which represented a just perceptible visual difference. The colors within these points were considered a match to the standard, while the colors outside were not considered a match. He repeated this experiment with a number of different colors and plotted the results on the chromaticity diagram. He discovered that instead of circles of equal radius around each of the standard colors, the points representing visually perceptible color differences actually formed ellipses of varying sizes. This demonstrated that equal visual differences are not always represented by equal distances. In other words, the chromaticity diagram is not uniform from a perception standpoint. Non-uniformity means that the distance for a visibly perceptible unit of color difference for a green standard is mathematically larger than the distance of a similar color difference for a red standard. Both are larger than that of a blue standard. Using the chromaticity diagram to determine measurements for color difference will therefore produce results that vary depending on the hue of the color being compared. While the measurements are correct, the mathematical interpretation of these measurements does not agree with our perceptions. A more visually uniform system is needed.